strategies to modulate cell functionality and to combat infections. A new perspectives to old challenges. Organized by Department of Microbiology, Ramakrishna Vivekananda Centenary College, Bangalore. We are very fortunate to have Dr. Bikramjit Basu, Professor, Material Research Center and Center for Biosystems in Science and Engineering, Indian Institute of Science, Bangalore, for today's seminar. We are very grateful to you, sir, for giving us time from your busy schedule, for sharing your valuable insights, views with our students. I would now kindly request our revered Principal Maharaji to felicitate Dr. Basu. And I'd like to request our revered Principal Maharaji, Swami Kamalasthanandaji, to kindly give his welcome address.
Most respected Professor Bikramjit Basu, the Professor of Material Science Research Center and Center of Biosystems, Science and Engineering, was IISc, Bangalore. And today we are very happy to organize this type of seminar. And I tell that this type of seminar is a special flavor. I think this will give some special flavor if you feel. Because sir actually afterwards my colleague will actually introduce him. Sir actually the students of metallurgy engineering, and now he is a great scientist in engineering of biosynthesis, biosystem synthesis. So such a vast journey, and for this purpose he is recognized all over India in a special scientist. And so his topic of discussion is not physics, not chemistry, not biology, not mathematics or so and so, but a special type of application of science in today's world, in today's different types of field, what we can read in different way. We read protein, we read enzyme, we read this chemistry, this reaction, that reaction, and also this type of metallic bond. So many things we have read. But how it will be applicable and how it can be transferable. And in this way, the new division, translational research, is a new division, and he is also a leading one, scientist. So you are, I think, you will be fortunate when you understand that sir actually delivered a talk before you and interact with you. And if now you can understand it good, but when afterwards you can understand, it is a privilege to you because sir is a very great scientist in this translational research. It is a different type of and emerging field which is required, and all over the world actually scientists are very much involved in this type of research. So we are very happy to have in this very beautiful afternoon. In spite of very busy schedule, he come here for a short visit and short engagement. In spite of that, I invited him, and he today, all in spite of his all constant, he came over here from his other type of or other type of engagement, though he has engaged different type of engagement. So on behalf of this college, on behalf of all faculty and students, staff member, I express my heartiest regards and welcome to Professor Basu on behalf of this college and on behalf of his presence in this college. And I tell all the students, please listen and interact with him wholeheartedly. Whatever you can learn, you can interact, and afterwards you can understand that this topic and his deliberation, how it will helpful in your this pursuit of science, this type of study and research. So, with these few words, I express my heartiest regard once again to Professor Basu and also welcome all the students and faculty members in this beautiful afternoon and all we enjoy this seminar. Thanks all of you. Thank you.
So before we begin, I'd like to request Dr. Sujoy Pal, Department of Microbiology and Distribution, Biochemical Center College, Rohra, to introduce our speaker to the audience.
Professor Bikramjit Basu has been pursuing research on multifunctional ceramics over more than a decade and successfully engineered biomaterials and impact areas such as healthcare, renewable energy, and aerospace. The knowledge and experience required to work in this interdisciplinary independent research career and materialistic science. Her research and related contributions have led to 260 research articles, 80 review papers, 5 textbooks, 10 book chapters, and 8,900 citations. His work focused on the end-to-end integrations and manufacturing, life cycles, and biomedicinal implements in materials that lead to interactions and new technologies. His academic appointments in now uh, honorary professor of School of Materials, University of Manchester, UK, but he is presence of various prestigious institutes. And 2021 last was the guest professor of Bonn University of Technology in China. He's also he has the 2018 to present. He is the vice president in Society of Biomaterials and Artificial Organs in India, and he also gave various prestigious awards. And there's a long list in here, but I just says about 2013, he got Chancellor Bhatnagar Award from the Council of Scientific and Institutional Research of Government of India. And 2018, he got the Global Ambassador Award in the American Ceramic Society of USA. And there is a long list of these awards, and we are very much fortunate to about Dr. Basu to present here. Thank you.
We are very glad, sir, that you are among us today. We now request Dr. Bikramjit Basu to deliver his speech. He will speak about bioengineering-based strategies to modulate cell functionality and to combat infections, new perspectives or challenges.
So good afternoon, everybody. First of all, I would like to express my sincere thanks and gratitude to respected Principal Maharaj Swami Kamalastanandaji for inviting me to visit our Ramakrishna Mission. This is the first time I'm fortunate to visit this great institution. I am associated with Bangalore Ramakrishna Mission for last few years, and I visited Bangalore Ramakrishna Mission. I also like very much the atmosphere, very peaceful atmosphere and serenity of Ramakrishna Mission in general. And in particular, I also believe in the philosophy of Swami Ramakrishna there. So, having said that, this is one of the outreach activities that I have been doing for last several years to visit different institutes across India to excite the young minds in the interdisciplinary field of bioengineering, which is at the intersection of biological sciences and engineering sciences. So what I'm going to do today, you'll notice in next 45 minutes or 1 hour, in the part 1 of my lecture, the bioengineering-based strategies to modulate cell functionality, to cell functionality, new perspectives to old challenges. Okay, so this is the first part of my talk, and in the second part I will be delivering. And in the second part, I'll be delivering the bioengineering-based strategies to combat infections. So what I'm going to show you in the next 45 minutes is that we have been working we have been pursuing this research at IIT Kanpur first when I was a faculty at the Indian Institute of Technology Kanpur. And from 2011, when I moved to Indian Institute of Science, I continued the same thing. So first few slides I will be introducing you. So first few slides I'll be introducing you to the the, uh, the the great academic universities of India, and most of you know that we have around 23 IITs in different states of India. Then in addition we have Indian Institute of Science, Education and Research, like nearest one is IISc Kolkata, that also came up very recently. So altogether we have 23 Indians of Technology, 7 Institute of Science and Indians of Science, Education and Research, and 1 NISA, that is the National Institute of Science, Education and Research. Now, despite having so many IITs and ISA, but we have only one Indian Institute of Science, and that is located in Bangalore. It was established in 1909. And then Dr. C.V. Raman, the Nobel laureate, was the first Indian director. And this idea was conceived in 1892 by Jamsetji Tata, and Swami Vivekananda actually gave this very special idea that India should have an institution of higher learning which will be only devoted to scientific research, and this scientific research should be at the global standard. And that was Swami Vivekananda's vision, which was translated to reality by funding from Jamsetji Tata. And this Maharaja of Mysore gave the land at the heart of Bangalore. And what you see is this was the building, the heritage building, the main administrative building of Indian Institute of Science. And this particular institute, why I'm showing you all these slides, you should be knowing that little bit history of some of the very famous institutes in India. This institute has not only produced thousands of scientists over last 100 years of its existence, but it has also produced some of the leaders who have really shaped different scientific fields in the country. And who are those leaders? You can see Sir C.V. Raman, Professor Homi Bhabha. He was a former faculty member of IISc, and you know Bhabha Atomic Research Center, right, in Bombay. Then you know Bikram Sarabhai, he founded the India Space Research, and you know there is Bikram Sarabhai Space Center in Trivandrum. Then Sir J.C. Ghosh, he was the first director of IIT Kharagpur, so you know Gyanachandra Ghosh. Then Professor Satish Dhawan, he was the chairman of Indian Space Research Organization, and Professor C.N. Rao, who is in Bharat Ratna, you know, and he has started this Jawaharlal Nehru Center for Advanced Scientific Research that you know some of you may like to choose in the years to come. So this was IISc Bangalore in the year 1909 when it was first established, this building. As you see, there is nothing in Bangalore city at that time point. And what you see now, it is 2022 Indian Science, and you can see that the signature of modern city, like you know many high-rise buildings and so on. Now, if you look at the Indian Science, their academic environment or academic ecosystem is can be divided into a number of divisions, like mechanical sciences divisions, and you can see most of the departments like materials engineering, aerospace engineering, civil engineering, chemical engineering, they are all part of this mechanical sciences division. Then we have physical and mathematical sciences, and under that you have mathematics, physics, and high energy physics. Then we have electrical engineering, computer sciences, like electrical engineering, computer science, and automation. Then we have chemical sciences. I belong to the chemical sciences. Then we have materials research center and inorganic and physical chemistry. Then we have biological sciences division, and then this is the molecular and reproductive development genetics, molecular biophysics, microbiology and cell biology, ecological sciences, and so on. And this is a newly created division that's known as interdisciplinary sciences. So interdisciplinary sciences essentially means there is no barrier between two disciplines. So it is all mixed up, or it is also in United States they often call it the convergent science. So we have this biosystem science and engineering. So it is not biology, it is not engineering, but it is the integration or adaptation of the biological concepts to engineering systems, or it is the adaptation of engineering concepts to biological systems, whatever way you can essentially define. Then you have cyber physical systems, management studies, and so on. So if you look at all the list of these different departments and centers, all together, we have 42 departments and centers at Indian Science Bangalore. I have visited many universities across the world, but I have very, with the exception of some of the American universities, many universities around the world, they may not be having so many different disciplines spanning across all the disciplines of science and engineering. And in terms of the different rank and in terms of the different numbers, what you see here, that IISc has around 5,000 students, 400 PhDs graduated per year, close to 3,000 publications, research papers, or peer-reviewed research papers per year, at least in 2021. And in all kind of rankings, Indian Science remain number one, whether it is a NIRF ranking, whether it's the World University ranking, whether it's QS University rankings, and the most recent ranking, if you see, citations per faculty, like when you publish papers, how each paper gets cited by researchers around the world. And in terms of these citations per faculty, IISc was ranked number one in the world. So it is in 2022. So these are like all the kind of rankings I can see. Oops. So in that, so it is selected by the Government of India as an institution of eminence in 2018, and this provides a lot of autonomy for this for the administration's academic administration. So what is new at Indian Science that you might have read in the newspaper? It is that we are going to have a medical school, and it will have a 500-bed hospitals which will have clinicians on campus, And there is a special degree that we'll be offering that is called MD PhD program. You must have heard MBBS program. Then there is a MD program. But hardly you find in India that many clinicians they have a PhD degree. So we'll have the MD PhD program, and this is the Bharti Pathsarathy Hospital. It is going to be functional in the year 2023, that is next year. So this is the new initiative that Indian Institute of Science we have taken, and this is the research and innovation facilities. So what it's going to do, it will allow us to pursue research to to address various unmet clinical needs in the area of diabetes and metabolic disorder, cancer, integrative medicine, infectious disease vaccines, and neurodegenerative diseases. Now, this is also, there is many other possibilities that it will allow. You must have heard of artificial intelligence, AI. So artificial intelligence enhanced healthcare, that is AIEH, that is also another one program that we are going to launch, and digital health is another one. Third is the biomedical devices systems, where our interest will be more for translation to products, like it will go to healthcare practice. All right. Now, having said that, I will now start my research. So this is what I'm going to show you is that we develop materials. We are engineer by profession. We develop materials, and what is biomaterials or what is bioceramics or biopolymers? That means we are developing synthetic materials, synthetic ceramics materials, which are expected to interact with the key components of the living system. And key components of the living system is proteins, blood, cells, and tissues. And you can see how the protein structure looks like. This is the typical eukaryotic cells. But at the same time, I have shown you bacteria, that is prokaryotic cell. So we want cells to adhere on the material, but we don't want bacteria to adhere on the material. You understand the significance? Why we don't want bacteria to adhere on the material? Because if the bacteria colonize on the materials, then it will cause infection, right? So we don't like bacteria to be attached, bacteria to grow on the material surface, but at the same time, we want cells to attach, cells to adhere, cells to grow, cells to proliferate on the material surfaces, okay? So this is the fundamental principle. Now, second one is the Rpc, red blood cells. Now, red blood cells is one of the cellular component of the human blood, right? Red blood cells have a very characteristic donut shape. We don't want that shape to be distorted. Red blood cells will be damaged if it comes in contact with the blood. And at the same time, the platelet, the same story is true for platelets also. Now, as I said in the last slide, that we want cells to grow on the material surfaces, but the natural question that should come to your mind, so this is a synthetic material, right? Thatched surface. Now, what you see, when a cell will try to interact on the material surfaces, then what will happen? Cells have certain cell surface receptors on the cell surfaces. And what you see in these geometric objects like triangle, circle, square, and so on, these are like different protein molecules, macromolecules, that is absorbed on the material surfaces. So essentially, cell-material interaction is nothing but cell surface receptor and these proteins which are absorbed on the material surface. Now it's an interesting thing. What you notice here, this is a spherical object and this is a flat surface. Now, from the very fundamental physics point of view, you know the stability of any spherical object would be much more if the spherical object deforms to some extent, so that it will have much more number of contact points or much more number of interaction points with the proteins which are absorbed on the material surface. And that is what exactly happens. And then final step, you can see this cell which is elastically compliant, because cells have, what are the structure in the cells? Cells have actin filaments, there's cytoskeleton, there's a microtubules, all these are like essentially proteinaceous structure. So this proteinaceous structure essentially means their elastic stiffness is very low, and that's why we call elastically compliant material. So if this elastically compliant material, it can stretch. So the cells can stretch on the material surface, And this stretching is possible because of these actin filaments. They can also stretch themselves, and as a result, these cells can expand. And you can see these red dots on the material surface. You can consider it as sticking points, but this is essentially focal adhesion complexes. So each of these focal adhesion complexes, like each of these red dots, essentially consists of a number of these interaction points of these particular cell surface receptors and the proteins which are absorbed. So what are the different aspects or what are the different attributes that influences the way the cell will interact with the materials? It is the stiffness, that means elastic modulus, substrate topography, that whether it is rough or whether there is fibrous-like structure and so on, substrate wettability, and biochemical growth factors. Now, this is the biochemical growth factors. This is the domain of biologists, while biologists use the biochemical growth factors to modulate the cell functionality, to modulate the way cells will behave, cells will behave. So, essentially, what we do is that I'll just skip some of the slides, then we'll go to that. Now, there is in the human body, there are different tissues and organs are there, and some of them are electroactive tissues like brain, skeletal muscle tissue, and also heart. So these are the electroactive tissues, and there is also mechanosensitive tissues like bone, ligament, and tendon. So what I'm going to show that what you have seen in the last one or two slides, that I was describing that how cells will interact with the material surfaces. So I was trying to show you that whether this cell surface or the way the cells will be cells can be stimulated on the material surfaces if the material surface can have positively charged or material surface can have a negatively charged. Then you can introduce the space charges in the materials. Then another way that I'm going to show you with different examples is that electrical stimulation. Electrical stimulation means when the cells are adhering to the material surfaces, you essentially apply the electrical stimulation, and the electrical stimulation follows like that. You have a cell incubation period, then you have electrical field, you can apply, then you give this electrical field for T1 time. Now T1 time can be few minutes to, like 2 to 5 minutes to 10 minutes. Then you can switch it off the electrical stimulation. Then you give the cells, then you don't apply the electrical stimulation for T2 time. Again you repeat for the T1 time. And this regular intermittent delivery of electrical stimulation, if you give, we have shown that it can essentially modulate the cell functionality. Now, what is the cell functionality? I can show you in the next. Many of you have heard the stem cells. What is the stem cells? Stem cells are essentially unspecialized cells which have the potential to divide, at the same time which have the potential to differentiate to different cell types. And what are the different cell types? Like I have shown you here, it can go to the fat cells, adipocytes, it can go to chondrocytes, that is cartilage cells, it can differentiate osteoblast, osteo means bone, osteoblast means bone forming cells, then we have muscle cells, and we have neurons. So mesenchymal stem cells can be differentiated to all these kind of cells, all these different cell types. So I repeat, stem cells on its own do not have any specific function. However, all the cell types have the different functions because neurons, you know that neurons is part of the central nervous system and so on. So what we are going to show you is that these stem cells, when you grow on a particular substrate, these stem cells, you can use the chemical or biological factors like growth factors, but that has potentially long-term negative impact on the physiology of the organism. So in order to avoid that long-term negative aspect, what we are going to show that when you grow the stem cells on the materials, if you give this kind of electrical stimulation, and I'm going to show you that how by tailoring electrical stimulation you can differentiate the stem cells to either osteoblast or to muscle cells or to neuron cells by carefully tailoring the electrical stimulation. So I have already mentioned about the stem cells. These first 3 points I have already mentioned. And those who are not from biological discipline, differentiation means differential gene expression. There are specific genes which have been upregulated, and therefore the cells will then have the ability to do certain specific functions which otherwise is not possible. And that I have mentioned. So yeah, how this stem cell differentiation can be modulated. You can use either mechanical or electrical stimuli, or soluble factors, or topography, or by substrate stiffness. And you can see the stem cells, when they will differentiate, they will have a specialized cells. And these specialized cells, if you see, that they are essentially, the nucleus will have different colors, so essentially shows that their genes are very different. Okay, one of the things about the stem cell differentiation means that when stem cells are committed to either to go through osteogenesis, like it will go through bone cells, or chondrogenesis, it will go through cartilage, or myogenesis, when it will go to muscles. This is not a single step differentiation. So it will go through different stages of this process of differentiation. There is something called commitment. Commitment means when stem cells are committed to go through osteogenesis, it will not go to chondrogenesis after some time. So it will be committed to go through this particular lineage. Ultimately, it will go to mature bone cells, which is known as osteocytes, right? So similarly, if it is committed to myogenesis, then it will be committed to myoblast, and then ultimately it will form the muscles. Okay. Now, when I said that cell stimulation is one of the important approaches of the bioengineering approaches, but you should be also aware that there is several other bioengineering approaches, like you can apply very local compression, or you can use magnetic tweezers or cyclic stretch. So these are the different ways that you can use the stem cell differentiation. So initially, when we started working on these materials, we tried to develop different kind of materials. So for example, this is hydroxyapatite and barium titanate. What is hydroxyapatite? Your bone, natural bone of all of us, like human beings, it is made up of 2 principal components. One is called collagen and another is called hydroxyapatite. So collagen is an organic component and hydroxyapatite is an inorganic component. Inorganic means it is a synthetic compound, hydroxyapatite. And barium titanate, calcium titanate, if you are not from materials background or chemistry background, barium titanate, calcium titanate, they are like certain piezoelectric materials or electroactive materials. If you add this barium titanate, calcium titanate, you can make the materials more electroactive. So these are essentially called elastically stiff substrate. Now there are also elastically compliant substrates are there, like carbon-based substrate. PANI essentially means polyaniline. Then there is ECM decorated PANI. Then there is a PANI polyaniline and gold nanoparticle composites, and on which you can grow stem cells. And if you grow stem cells, you can apply the electrical stimulation, and you can see that how the electrical stimulation will work. So the first one that we have done the experiments, and when we have not given the electrical stimulation, that's why it is crossed. So you will see that this is the hydroxyapatite, which has conductivity of 10 to the minus 14 Siemens per centimeter, and it goes all the way up to 100% calcium titanate, their conductivity 10 to the minus 3 Siemens per centimeter. Now, if you grow the stem cells, and then what you see that myoblast cells, once they start forming, these myoblast cells are aligned. So this is a fluorescence microscopy images. Many of you may have done fluorescence microscopy in the lab, so they know how the fluorescence microscopy works. And if you look at the myoblast density, as I said, myoblast is made muscle cells, and if it is plotted with this conductivity of the substrate, you will see that how substrate conductivity modulate the more and more number of myoblasts and myotube formation in vitro. So this is the work which is published in Biomaterials. Then we have done some experiments as part of one of my PhD students, Silpi joined at IIT Kanpur, where we have grown stem cells on carbon-based substrate. So this is the carbon-based substrates, which is essentially what as electrode, and then we grow the cells on this carbon-based substrate, and then we'll see that how stem cells can be, how neural cells can be activated with the increase of the electric field strength, and neural outgrowth is maximum at this particular electric field strength. So essentially, what is the message that you are getting? That all this electrical stimulation, you need to essentially optimize in order to get the maximum potential of the electrical field towards stem cell functionality modulation. And what is the neurite outgrowth? You can see this particular neurite that they are growing, and you can see these neurites are growing like a very long appendages, and that will lead to the more neurite outgrowth. Then comes polyaniline. Now, polyaniline is one of the electroactive polymer. Now, what we have done, we have brought hydrochloric acid to polyaniline. Now, depending on the extent of hydrochloric acid dopant to polyaniline, you like there is a -5 molar to there is a -1 molar. And when you have grown stem cells, you can see at certain hydrochloric acid doping, the cell morphology or cell phenotype is changed. You can see here, it's a more like neurite projections. And this more neurite projections is very clear on the way these stem cells are grown on this particular polyaniline. Now, what you notice here, when cell density and then cellular extension is plotted against open concentrated molar HCl, so that neurite outgrowth or cell density goes down. That means less and less a number of cells are there on the substrate, but at the same time you can see that cellular extension is increased. What it means that if you give this electrical stimulation in a very regular period, you can essentially allow the more cellular extension process to take place. So this is the case. You can see that there is a nestin is one of the nestin and beta-3-tubulin as neural markers, and these both the nestin and beta-3-tubulin markers are expressed in these stem cells when they are grown on this particular polyaniline substrate, and at the same time you can see large phylopodial extension. So whenever this phylopodial extension is extremely large, it will remind you of the neuron. If you look at the neuron classical picture, it is a long tail, right? So this kind of structure very much indicative the stem cells are undergoing neurogenesis. But the ultimate proof that will come that when you use that specific genes, as I said, beta-tubulin, nestin, and neurofilament are three specific markers which are upregulated when compared to GAPDH. GAPDH is the housekeeping gene. So this gene expression changes is very important. And in summarizing this part of the work, what you see here, you grow HMSCs, the human mesenchymal stem cells, you give electrical stimulation, and this stimulation is given in a very regular intermittent pattern. Then you allow human mesenchymal stem cells to undergo neurogenesis towards neural-like cells. And that has been confirmed by our analysis of the gene expression for different specific genes. Now, last part of this stem cell differentiation study, you'll see the stem cells. What we are going to show, this is the important concept. Before the lecture, somebody was telling that that my lecture may be very different from chemistry or something. But what you see here, that we are applying all the materials principle, chemistry principle, biological principles all together in this bioengineering platform. So what we have manipulated now stem cells, you will see this is the gold nanoparticles. And this gold nanoparticle synthesis is not anymore rocket science, because there are several methods. That one of the methods that we apply is the citrate cap method. And these gold nanoparticles, you can see these are negatively charged on the particle surfaces, right? Appendages. So what we have done, we have incubated the stem cells in gold nanoparticle loaded solution, and because of the extremely fine size of the gold nanoparticles, like less than 10 or 20 nanometer, they can be internalized into the stem cells. Listen to me very carefully what I'm saying. I'm saying you want more nanoparticles to be internalized into the stem cells. And therefore, you can see that more nanoparticles are sitting inside the cells now. And then we have grown on a substrate which is essentially copper slip, then polyaniline, then gold nanoparticle, then polyaniline, then gold nanoparticle. It is known as layer by layer deposition of gold nanoparticles sequentially on the polyaniline. So what the cells are seeing, cells have now internalized gold nanoparticles here. Cells also are sitting on the gold nanoparticle loaded layer. Now, when we apply the electric field, what will happen? This particular gold nanoparticle will work like intracellular actuator, and these particular gold nanoparticles which are sitting outside the cells, that will act as an extracellular actuator, right? So this, by applying the electric field, now you are trying to manipulate the stem cell functionality changes. Okay? So this is a basic concept that I am trying to propose in this particular slide. Now, this is about chemistry, as I said. So it is a citrate reduction method. You start with HAUCl4 and trisodium citrate. So this is the precursors. And these precursors, you allow them to react. The ultimate result is these gold nanoparticles, and by treating them with different acids, like one is a martactosuccinic acid, you can have negative charges, and in otherwise you can have positive charges when you treat them polyl-lysine. Now, two types of electrical stimulation I am giving. This is interesting. You just listen to me very, very carefully. In one case, we are giving the DC, direct current stimulation, not like any pulsar type stimulation. In another cases, we are giving pulse direct current stimulation. So we are putting the current on for few milliseconds, off, again on, again off. So you can see this goes on like 100 milliseconds, 100 millivolt current on, then 900 millisecond off, then 100 millisecond on, 900 milliseconds off. And if you give to the same system in gold nanoparticle, now you'll see what will happen next. Now, as I said before, gold nanoparticles, so this is the bar, micron bar is 50 nanometer. So individual size is around 10 to 20 nanometer. So that is a very fine particles. And this is like internalized into the cells. Now, when you see what happens when you give the electrical stimulation, then the electrical stimulation are given very, very thoughtful manner. We allow the cells to settle down, and there's a pre-induction period. Then we allow cellular uptake of the gold nanoparticles. Then we apply from day 3 to day 14 is the intermittent electric field stimulation. And this part you can see electrical stimulation is given for 15 minutes, then 24 hours rest period, 15 minutes, then another 24 hours rest period. It will go for a few days, up to 14 days in culture. In a pulse electrical stimulation, this 15 minutes time is broken into 100 milliseconds on, 900 milliseconds off. So this was done after so many experiments, and what you see here, that there is no dead cells, like this is a lighted imaging, everything is more green, and there's only one or two small dots which are essentially dead cells. Now, these gold nanoparticles, when you are internalizing into the cells, you see that if you do it for gold nanoparticles per cell, that number is very large, even after 1 day in time. So therefore, this 1 day is good enough for gold nanoparticles. Now, when you give this DC stimulation, interesting thing happens. You can see the stem cells are changing the phenotype. They are very significant morphological changes, very long appendages, and this actually reminds you like a neurite outgrowth. If you look at the one single cell here, you can see there's a long branches and there are also shorter branches here. We try to quantify in terms of the cell morphological changes, in terms of the length of the neurite-like outgrowth, what is the individual branch length, and what is the number of branch points. So essentially, this is clear indication that stem cells have undergone more neurogenesis kind of thing. Now this one, when we have included core nanoparticle and bilayer composites and on which stem cells are grown, you can see how this branch length changes and what is the length of the neurite also that changes at different time periods. So we have also done all the specific genes. One of the genes is nestin, neurofilament, beta-3 tubulin, and in all these cases, nestin, neurofilament, beta-3 tubulin, we have found out that when the cells are grown on layer by layer deposited core nanoparticles, then you can see that mRNA expression is much more. So what it shows that cells have indeed undergone neurogenesis towards this more beta-3 tubulin, and you can see this DC electric field stimulation, this beta-3 tubulin expression is much more when your cells are growing on the layer by layer deposited substrate. Now, interestingly, what you see here in this slide, when you are given this kind of stimulation, not like this electric field, but the pulse electric field stimulation, what I have shown you a few slides back here. So if you look at this particular pulse electric field stimulation, when you have given this stimulation to the stem cells, now the stem cells, they have changed their morphology, but now the morphology is very different. You can see that there is more tube-like structure. You can see this is the tube-like structure, and this tube-like morphology has lesser cell shape index. What is the cell shape index? Area of the cell with the perimeter square. And there is also nucleus elongation factor. You can see nucleus are not anymore circular or rounded. Nucleus is more elongated. You can see this is the nucleus is elongated. Here also you can see nucleus is elongated. Now, nucleus elongation factor and cell shape factor, they are more when you give this electric field stimulation at 1 Hz frequency. And what is 1 Hz frequency? Your heart beats at 72 pulse per second minute, right? So this corresponds to essentially heartbeat rate, and this pulse electric field stimulation essentially shows that 3 different genes like cardiac troponin, T, actinin 2, and beta 4. So these are the 3 specific genes which are essentially upregulated with the presence of the electric field, and however, they are not overexpressed without electric field. So essentially, it clearly shows electric field has specific role to play in terms of this particular cardiomyogenesis. That means that these stem cells undergo more like cardiac type cells. So we have done a lot more analysis, but I'm going to summarize this part of the work by showing you this slide. If you give this electric field, stem cells go through more neurogenesis. If you give pulse electric field stimulation, stem cells undergo more cardiomyogenesis. So in one case it is neural-like cells, in another case it is the cardiac-like cells. So this one is possible because during the cell proliferation process, there is a premature cell cycle arrest at G0, G1, and that triggers very early differentiation of intelligence. Okay, so yeah, this part of the work what I'm going to show you. Now, some of you might have heard about the lab on a chip or organ on a chip. What is the meaning of lab on a chip or organ on a chip? Like you have some microfluidic devices, which is bioengineered devices, which you can use to precisely control these stem cell fate processes towards specific differentiation. So I'm going to show you now that within these microfluidic devices, if you give shear, then the stem cells, if you take, they will not only retain the stemness, but also it will allow osteogenesis process to take place. And this is the 2 composites that we have shown. One is the ultraviolet polyethylene, and then I'm going to show you, yeah. Yeah, so when you grow these stem cells in this shear, you will see that there is several aspects that you can see. One, the ultra-oil molecular polyethylene, this is one of the biomaterials, and when you have added this multiple carbon nanotube, then you can see that stem cells, they are growing very, very significantly, and there's a left-right parallelity that is also, you can see very nicely. Then there's ultra-oil polyethylene, when we add this hydroxyapatite to it, then also stem cells will grow signs, there are specific signs, and these signs essentially tells you that they are going through more like a bone-like cells. That's why it is called osteogenesis. And this is a very clear evidence that this is called left-right parallelity, and that is a specific attributes that stem cells. You can see this shear essentially allow that specific rotation within the cytoskeleton of the actin filament. And this is something very special attributes of the stem cells, and that is essentially tells you that how stem cells can be, stem cell differentiation can be modulated. Then there is another aspect in the stem cells that is called transdifferentiation. Transdifferentiation means we are growing now muscle cells with human mesenchymal stem cells. Now, what is muscle cells? Muscle cells is already one matured cells, right? Because stem cells can differentiate to neuromyogenesis and to muscle cells. Now, we have shown when you give this electric field stimulation and if you co-culture muscle cells with muscle cells with human mesenchymal stem cells, what will happen? It will undergo transdifferentiation, means muscle cells will first go back to stem cells, and both stem cells and differentiated, trans-differentiated muscle cells, they can go through another path through what is called neurogenesis. So this is kind of very, very interesting observation that we have made. So what we have done, we have grown this cell channel. Here we have grown this co-culture. Then we have put two electrodes in the two opposite sides, and then we have given this kind of electrical stimulation for 20 hours. And it's different types of days in culture, up to 9 days in culture. And after giving this electric field, like 3.5 microvolt per millimeter, we have seen that how muscle cells they grow trans-differentiated. And if you see the cell-to-cell communication, this is a beautiful process microscopic images and different stained images. You can see very large elongations, right? Large phylopodial extension. And this large phylopodial extension essentially tells you that this co-culture cell population must have gone through the neural cell types. So I'll just go through some of the more neural-like morphology, and more neural-like morphology. You can see there's a very large, long, elongated structure, and that will go through, that will go through neurogenesis. Okay, so now another interesting thing in this particular case, you can see. So, okay, so yeah, so this is calcium ion, and so there are three ion channels in the cells. One is the sodium potassium channel, and one is the calcium channel. And both the channels are voltage gated channels in the cells. So when you give this electric field stimulation, these essentially calcium ions they get activated, these calcium ion channels, and you can see below how these again the calcium channels are activated. In the case of these intracellular communications, you can see that this particular Pascal multiplier study essentially shows how voltage-gated calcium channels are also getting excited about this particular electric field stimulation. So what I'm not going to show you is in this particular presentation that we have done a lot more electrical stimulation on the hydroxyapatite-based electroactive material, where we have shown if you grow human mesenchymal stem cells, if you give this electric field stimulation for 10 minutes up to 4 weeks in culture, essentially you can allow the differentiation of human mesenchymal stem cells to pre-osteoblast and then osteoblast-like cells. So osteoblast means bone forming. So essentially it is the clear case of osteogenesis. Now what happens to the stem cells when they are giving electric stimulation? So what I'm showing here, there is something called mechanotransduction effect. That means cell signaling processes are mechanically stimulated, and this mechanical stimulation is possible because when you apply the electric field, then if you solve certain Poisson-Boltzmann equations, then what you see, you can get the stress level. You can find out what are the different components of the Maxwell stresses in the cells, And if you compute, then you will find out that these stresses are of the order of micropascal level. Micropascal means 10 to the power minus 6 Newton per meter square. So this is very small stress level, and this cross stress level is good enough to upregulate certain gene expression in these materials. Okay, so I'd like to acknowledge the funding agencies, the Department of Science and Technologies, then CSIR, Department of Biotechnology, Indo Science and Technology Forum, and so on. So we'll have 5 or 10 minutes break now, and then I'll start the part 2 of my lecture. Okay.